Hello, I'm Dr. Gonzalez, and today in this video we're talking about the following topics.
In our previous video, we were talking about the kidney and we were talking about the general anatomy of the kidneys. In this video, we're going to concentrate on the nephrons and we're going to go over some of the general physiology. So for this video, let's start with the nephrons. First thing that you need to know is that your nephrons, some of the things that they do is filtration, reabsorption, and secretion. In each one of these parts of the nephron, these steps are going to be happening. So for example, the first step is going to be filtration from blood plasma into the actual nephron in the area of the glomerulus. The second step is going to be tubular reabsorption from fluid into blood. And the third step is going to be tubular secretion from blood into fluid. In tubular reabsorption, 99% of water and solids are reabsorbed. And in tubular secretion, waste, drugs, and excess ions, it's exactly what we are secreting. Now, in the glomerular filtration, or in this area right here, this is going to be driven by blood pressure. It's going to be also opposed by capsular hydrostatic pressure, which is the hydrostatic pressure against the membrane, and blood colloid osmotic pressure, which is due to the presence of proteins that opposes that filtration. Water and small molecules will move out of the glomerulus in that direction. And in one day, just FYI, 150 to 180 liters of water pass out into the glomerular traction. In glomerular filtration, like I said before, it's going to be driven by several forces. For example, one of the forces is going to be glomerular blood hydrostatic pressure, which is 55 millimeters of mercury. The second one is going to be capsular hydrostatic pressure, which is 15 millimeters of mercury. And then blood colloid osmotic pressure, which is 30 millimeters of mercury. That being said, we can calculate the net filtration pressure with these three forces. So the net filtration pressure is the glomerular blood hydrostatic pressure minus the capsular hydrostatic pressure minus the blood colloid osmotic pressure, and that gives you 10 millimeters of mercury.
Okay, so now it's time for a short discussion break.
Anatomically speaking, the renal corpuscle is also close to the juxtaglomerular apparatus. And in this case, the juxtaglomerular apparatus, the ascending loop, which is this area right here, contacts the afferent arteriole, which is this one right here, and it contains smooth muscle cells, we also known as the juxtaglomerular cells. The apparatus basically regulates blood pressure in the kidney in conjunction with the autonomic nervous system. So this is going to be your macula densa of juxtaglomerular complex, and these right here are going to be your mesangial cells. In order to understand what happens in each part of the nephron, we have to also understand the anatomy and physiology. So first of all, remember that in your nephron, this area right here is considered the proximal convoluted tubule. This area right here is considered the loop. This area right here is considered the distal convoluted tubule, and then this part is considered the collecting duct. So in each area, we're going to have different types of cells, and these cells are going to be absorbing or secreting different ions or water depending on the scenario. So in this drawing that I have right here, I'm just resembling each one of those parts. For example, the proximal convoluted tubule, the loop, distal convoluted tubule, and collecting duct. So this right here is resembling the lumen of the cells, right, inside the tubules. This right here represents the cells. This right here represents the extracellular fluid in this area right here. And then last but not least, the capillaries. So what I'm showing you here, it's how we move all the way from either the lumen through the cells, through the extracellular fluid into the capillary, or vice versa. So in the proximal convoluted tubule right here, you can observe how we have movement of water and movement of different ions such as chloride, potassium, calcium, and magnesium, as well as urea, either by diffusion and water, in the case of water, by osmosis. In the loop, we're going to have, for example, movement of sodium, potassium, chloride, calcium, and magnesium. Some of these are going to be moved through diffusion. Some of these, such as sodium, potassium, and chloride, are going to be moved thanks to this symporter, which is this right here, and it goes then through these sodium-potassium pump towards the capillary. Then in the distal convoluted tubule, again, we're going to have another symporter right here that moves sodium and chloride, and we're also moving calcium in between these cells going towards the capillary. So most of these areas we're going to have a lot of reabsorption, that's what I'm trying to say. And then, for example, in the collecting duct, which is the last part, we're going to have, for example, movement of sodium, potassium, water, of course. We're going to have these aquaporins right here, a lot of aquaporins actually. We're going to have bicarbonate, and then we are going to have also some reabsorption and secretion, in this case, of these sodium, potassium, water, right? Reabsorption of those inside the collecting duct. As you can see here, many of the transport mechanisms are going to be either primary active transport using ATP, like those sodium-potassium pumps, and then we're also going to have secondary active transport, which is driven by ions electrochemical gradient. In this case, we can have symporters that move substances in the same direction, and antiporters, which move substances in opposite directions. The last thing I'm going to go over in this video is over the Renin Angiotensin Aldosterone system, also known for short as the RAAS. So the RAAS starts whenever we have a decrease in blood volume and blood pressure. So in this case, now what happens is that the juxtaglomerular cells secrete renin in response to this blood volume and blood pressure decrease. Right after that, renin aids in the production of angiotensin, which is produced in the liver. And then last but not least, in these 4 steps, the first 4 steps, is that angiotensin-converting enzyme, or ACE for short, changes angiotensin 1 to angiotensin 2, which is the active form. This ACE is produced in the lungs. Once we have angiotensin 2 already ready to go, angiotensin 2 is going to help decrease the GFR by creating a vasoconstriction of the afferent arteriole. Then it enhances the sodium, chloride, and water absorption in the proximal convoluted tubules. Once that happens, this stimulates the adrenal cortex to release aldosterone, which stimulates the principal cells to reabsorb sodium and chloride and secrete potassium. Last but not least, once there's a release in aldosterone and we have reabsorption of sodium, chloride, and secretion of potassium, we're going to have absorption of water increasing, and this also increases the blood pressure and the volume too.
Well, that's it for this video. I hope you like it. Don't forget to subscribe, like, and share, and also comment if you have any questions or suggestions about this topic. And also, if you like this shirt, it's now available on my Etsy shop, Queen Mary Anatomy. That's it for today. Thank you for watching.